So let's get into Pyruvate Dehydrogenase Complex or PDC. As the name says, it's a complex enzyme. It is there in the mitochondrial matrix. It does the simple thing. What is the simple function this PDC does? It just simply breaks pyruvate into acetyl-CoA. That's it. That's what is the function of PDC. PDC simply oxidizes pyruvate, which is a 3-carbon molecule, into acetyl-CoA, which is a 2-carbon molecule. As you see here, look at the figure. All of you pay attention to this figure and count the number of carbons in pyruvate. We have number 1, number 2, number 3. Okay. And look at acetyl-CoA down here in the product. Number of carbons 1 and 2. SCOA, the C that you see in SCOA is not carbon, it is CoA. You know that, right? CoA, coenzyme A. That's not carbon. This is not carbon. That is coenzyme A. So acetyl-CoA has only 2 carbon. So 3-carbon pyruvate oxidized into 2-carbon acetyl-CoA. That means one of the carbon is released as carbon dioxide. And that is what is this carbon dioxide here. And that is a decarboxylation. Decarboxylation, you are removing carbon, right? And your pyruvate is oxidized to acetyl-CoA. That means something has to be reduced. Because when something undergoes oxidation, other thing has to be reduced. It's a coupled reaction. Whatever you remove electrons, it has to be placed somewhere. And where you are going to place it? You're going to place it on NAD. NAD takes those electrons and become NADHH plus. So NAD is reduced into NADH plus H plus. So you're going to get one NADHH plus there by oxidation of pyruvate into acetyl-CoA. So every molecule of pyruvate oxidized to acetyl-CoA, you're going to get 1 carbon dioxide and 1 NADH. How many pyruvates you have coming from glucose? We have 2 pyruvates. So that means 2 acetyl-CoAs. It means 2 NADH and 2 carbon dioxide. So out of 6 carbons of glucose, by the time it becomes acetyl-CoA, 2 acetyl-CoAs, already 2 carbons are removed. How many carbons left now? Only 4. 4 carbons in the form of 2 acetyl-CoA. 1 acetyl-CoA, 2 carbon, another acetyl-CoA, 2 carbon. So we have 4 carbons which are from glucose there in the form of acetyl-CoA now. And those carbons need to be removed. And that will happen in the TCA cycle. So right now in PDC, you are removing one of the carbon from pyruvate. Okay, so that carbon will go into, we are going to exhale it out. It's a gaseous molecule, it enters into blood and it will go to lungs and we exhale it. Okay, there is a mechanism for that. You will study that in hemoglobin lecture. Okay, so in order to do this simple function, that is oxidizing pyruvate into acetyl-CoA, this PDC, it needs 3 subunits. It has 3 subunits. It's a complex enzyme. If I break this PDC, say draw it something like this, I'm going to divide this into E1 subunit, E2 subunit, and E3 subunit. Each one of these subunits, they need a specific coenzymes, which are mostly with water-soluble vitamin derivatives. E1 subunit, as I written here, it needs TPP, thiamine pyrophosphate, that is vitamin B1 derivative, right? E2, it needs lipoic acid, which is not really a water-soluble vitamin, but then there is a pantothenate. The CoA part in acetyl-CoA, the CoA come from pantothenate. Okay, so pantothenate and lipoic acid are coenzymes for E2 subunit. And remember, this lipoic acid has SH2. It has got thiol group, SH2 groups. Okay, I'll come to that in arsenate toxicity. And then E3 subunit need FAD and NAD. Now you know FAD coming from riboflavin, NAD coming from niacin. So that means how many water-soluble vitamins participating in this in the PDC? Vitamin B1, vitamin B2, that is riboflavin, vitamin B3, niacin, vitamin B5, pantothenate, and lipoic acid. Okay, these are the requirements that you need to know. Okay, so in this overall reaction, pyruvate is oxidized and it is undergoing and it is releasing carbon dioxide. That's why combination of this, we call it as oxidative decarboxylation. Oxidation and decarboxylation. As you see, pyruvate is oxidized and also it is decarboxylated. So that is why it is referred as oxidative decarboxylation. The name of the enzyme is Pyruvate Dehydrogenase Complex. Okay. Anywhere else did you see this kind of names earlier? Where? No, complex. Dehydrogenase complex. Water soluble vitamins, vitamin B1, and 4 enzymes you need to remember. In that slide, there were 4 enzymes. Vitamin B1 slide. Go back to thiamine, vitamin B1, 4 functions of vitamin B1. There were 4 enzymes mentioned in this slide. One is oxidative decarboxylation, 1, 2, 3, 4. And then trans-ketolase, pentose phosphate pathway. That slide, remember that slide. Okay, so those are the enzymes which needs thiamine. Now in that oxidative decarboxylation that I have written there, I have written 3 enzyme names which ends as complex. And one of that is this pyruvate dehydrogenase complex. Other was alpha ketoglutarate dehydrogenase complex. You're going to see that in TCA cycle. And the third one was branched chain alpha keto acid dehydrogenase complex. So all these 3 enzymes, they are of same family. Like you all, UMHS family. Each are subunits, students. Okay. So I'm like a regulator or something. Anyway, so E1, E2, E3, this is the arrangement of all these 3 complexes. So all of them, all these 3 complex enzymes, they need TPP, they need FAD, they need NAD, they need pantothenate, they need lipoate. The requirement is same. Okay, the way they function also same. They all conduct oxidative decarboxylation. Only thing is this, their substrate is different. If it is the substrate is pyruvate, you have pyruvate dehydrogenase complex. If the substrate is alpha ketoglutarate, you have alpha ketoglutarate as a substrate. Alpha ketoglutarate dehydrogenase complex. If your substrate is branched chain alpha keto acids, your enzyme is branched chain alpha keto acid dehydrogenase complex. So substrates are different. Other than that, the way these enzymes function, the way they are arranged, it is all same. Okay. Why I am saying this is, if there is any issue or if there is any deficiency of NAD or say FAD or say pantothenate or say TPP, all 3 enzymes are affected, not necessarily only this. Okay. We will get into that. See, I have written those 3 enzyme names here. Oxidative decarboxylation. Make sure you know this. Now, everything I have put like blue color. Wherever you see blue color, important around it. Okay. Oxidative decarboxylation, 3 complex enzymes. Make sure you remember the names and also where they are present. Pyruvate oxidation, TCA cycle, branched chain amino acid metabolism. So these are the enzymes which have got similar requirements. They all have E1, E2, E3 subunit. Then I have written something else down here. Each one of these E1, E2, E3 subunit, these are protein subunits. That means they are coded by genes. You know, any protein, you need a messenger RNA for that, and then the protein synthesis goes on. So for E1 subunit, for E2 subunit, each one of them, they have their own separate genes. That means there is a gene, separate gene for E1 subunit of pyruvate dehydrogenase complex, separate gene for E1 subunit of alpha ketoglutarate. If you go on saying like that, say the same thing for E2 subunit. Each one of them have individual genes for E1 and E2 subunit only. For E3 subunit, they share same gene. That means same E3 gene will give E3 subunit for all three of them. That means it is super active. What it does, it's going to make more and more E3, and it is one is going to pyruvate dehydrogenase, other is going to alpha ketoglutarate, third is going to alpha ketoacid dehydrogenase. So same gene is coding for E3 subunit of all three complex enzymes. You got to remember that. Okay. What is the purpose? What is the application? Why it is so important? As I have written here, above three enzyme complexes have separate genes for even E2 subunits, but they share same E3 gene. What I'm going to do now is I'm going to mutate this E3 gene. Imagine that E3 gene is mutated. So what will happen? What happens to these three enzymes? All of them are down. If E3 gene is mutated, all of them are down because it is sharing the protein subunit for all three of them. All three of them are down. So that means when E3 is down, so pyruvate dehydrogenase complex not working, that means pyruvate builds up. This pyruvate which is building up, that eventually leads to, it will go into lactate. Yesterday we have seen, whenever pyruvate builds up, what happens? It has got an alternate way to go. It goes to lactate. So that's why there will be buildup of lactate. And if alpha ketoglutarate dehydrogenase is not functioning, glutarate dehydrogenase complex, alpha ketoglutarate will increase, which I have written here. And branched chain alpha ketoacid dehydrogenase complex also down. That means branched chain alpha keto acids will also increase. So when E3 subunit is mutated, you're going to see elevation of lactate because it is coming from pyruvate. You're going to see elevation of alpha ketoglutarate. You're going to see elevation of alpha keto acid. Remember, these are all alpha keto acids as such. Pyruvate is an alpha keto acid. So that is why, especially for third one, I have written branched chain alpha keto acids. They are coming from branched chain amino acids. What are all the three branched chain amino acids? Valine, leucine, isoleucine, maple syrup, urine disease. So it is connection. It's a very easy disorder to identify, at least in the exam. Because as soon as you see sweet odor or a burnt sugar odor, that's maple syrup. And your amino acids is branched chain amino acids and their derivatives, branched chain alpha keto acids. Okay. So these branched chain alpha keto acids, when they are elevated in the blood, they will appear in the urine because they are water soluble. So the body fluid, like whether it is urine or a sweat, it smells like burnt sugar or a maple syrup odor or a sweet odor. Okay, so that is one of the clue for you to recognize, okay, there is an issue with the branched chain amino acid metabolism. So this is what happens with E3 subunit gene mutation. Okay, remember that. So if you see all three of them together, think about all three are down, that means E3 must be mutated. That is how you should approach to these questions. Coming to the regulation. Yes. Alpha ketoglutarate is an alpha keto acid. It's an acid molecule. So overall, because of all three of them, patient will have metabolic acidosis. We don't have separate sign for this alpha ketoglutarate, but overall, patient will be in metabolic acidosis. Why? Because, lactic acid, alpha ketoglutarate, it's an acid, COO minus, and alpha keto acid. They are all acidic molecules. There will be metabolic acidosis and the consequences of metabolic acidosis. Coming to the regulation of PDC. PDC, what it does, it is oxidizing pyruvate into acetyl-CoA. And the product is acetyl-CoA here. And the co-product, as you see here, co-product is NADH. As you see, there is a NADH formation, acetyl-CoA formation. Whenever there is accumulation of NADH in the mitochondrial matrix, or whenever there is accumulation of acetyl-CoA, if acetyl-CoA not entirely getting into TCA cycle for oxidation, whenever these two molecules build up. So whenever these two molecules build up, when they accumulate, so what do you expect them to do to your pyruvate dehydrogenase complex? Feedback negative effect, is it not? When the product accumulates, it will have a feedback negative effect. That's a classic feedback mechanism. Product will have a negative effect on the key enzyme in the metabolic pathway. It's not like immediately it will have, only when it builds up to certain level, threshold, then only it will have a feedback negative effect. So same thing happens on pyruvate dehydrogenase complex. That's why if you see allosteric regulation, negative modulators are acetyl-CoA and NADH. Make sure you remember this. It's a very simple fact, but sometimes you may forget it. So always remember, when NADH builds up or acetyl-CoA builds up, it will have a negative effect on pyruvate dehydrogenase complex. Thereby, pyruvate is not going down into acetyl-CoA. So pyruvate starts to build up. When the pyruvate starts to build up, what will happen to the pyruvate? What happens to that pyruvate which is not undergoing oxidation in the mitochondrial matrix? It will go back into the cytoplasm and go into lactate. And that's why when there is accumulation of NADH, when there is accumulation of acetyl-CoA, you expect there can be increase in lactate. That is what happens when you are doing rigorous exercise. When you do rigorous exercise, you make so much of NADH, you won't be able to provide oxygen sufficiently to keep your electron transport chain in accordance to how much NADH is produced. So eventually electron transport chain gets saturated. NADH start to build up, and that NADH will have a negative effect on TCA cycle and this enzyme, and then start to build up pyruvate, and pyruvate goes to lactate. It takes time, right? So eventually, as you go on working out, like maybe an hour or something, you start feeling the pain. Okay, that's because these negative effects of the molecules on the enzymes and eventually building pyruvate into lactate. Okay, so that's why yesterday I was saying, if there is any issue with your electron transport chain, there is an inhibitor on electron transport chain, oxygen not sufficiently available, mitochondria is mutated, mitochondria is dysfunctional, or some issue with the TCA cycle not running properly, what do you expect? Lactic acidosis. Because pyruvate don't go entirely into acetyl-CoA into carbon dioxide. So any issue with the electron transport chain, any issue with your mitochondria, overall mitochondrial function, any issue with your TCA cycle, expect lactic acidosis. There are mitochondrial inherited conditions. You are going to study in genetics, mitochondrial diseases. Not yet covered, right? There are mitochondrial disorders, maternally inherited. Leber, hereditary optic neuropathy, MELAS, myoclonic epilepsy, lactic acidosis. So there are any mitochondrial issues, mitochondrial myopathies, lactic acidosis. Remember, electron transport chain affected. Okay, so keep that in mind. Coming to positive allosteric modulation. In order to understand this, look at what are the positive modulators there, and you know the answer for that. What is the positive modulator here? Let's begin with ADP. Your cell has a lot of ADPs. What is the energy status of your cell? Low energy. What cell has to do? Increase electron transport chain. In order to increase electron transport chain, what it needs? It needs NADH, it needs FADH, it needs oxygen. How do you get NADH and FADH? TCA cycle. How do you run TCA cycle? You need acetyl-CoA. Is it not? Without acetyl-CoA, you cannot run TCA cycle. How do you get acetyl-CoA? You got to run this reaction. So that's why ADP, what it does, it will keep this enzyme active. So whenever there is increase in ADP, it simply keeps pyruvate dehydrogenase complex active and oxidize more and more pyruvate into acetyl-CoA, and acetyl-CoA enter into TCA cycle, because the same ADP will have a positive effect on one of the key enzyme in TCA cycle also, thereby keep TCA cycle high. So more ADP means your pyruvate oxidation into acetyl-CoA and later acetyl-CoA into carbon dioxide increases. That's it. Because you need energy, cell needs energy. More ADP means less ATP. You run the cycle faster, metabolism increases, and that is what is a positive modulation. Which is your positive modulator of glycolysis in extrahepatic tissues? What is the glycolytic most regulated enzyme in glycolysis? PFK1. In the extrahepatic tissue, which is the positive modulator of PFK1? AMP. Remember AMP? Whereas in liver, it is fructose 2,6-bisphosphate, but in all other tissues, it is AMP. And what is the indication of energy status of cell when there is more AMP? Low energy. That's why it is keeping glycolysis high, right? So lack of ATP means you will have more ADP and you will have more AMP. More ADP, more AMP means, it's a hunger signal. So metabolism goes faster. That's why AMP is a positive modulator on glycolysis PFK1 and ADP is a positive modulator on pyruvate dehydrogenase complex. Eventually, metabolism goes faster. Now, let's come to calcium. When you are working out in the gym or you are running on the, wherever you are running, what happens to calcium levels in the muscle? You go up. Muscle contraction means you need calcium. Calcium has to rise. Sarcoplasmic reticulum, calcium release, actin myosin contraction, more calcium. More calcium means muscle is contracting. When the muscle contracts, it also need to relax. It's in and out. Contraction, relaxation. Contraction, relaxation. That is actin myosin. For relaxation, what you need? You need ATP. To break the contraction, you need ATP. Otherwise, it will be there in contracted all the time. It becomes rigor mortis. Rigor mortis after death, what happens? For a couple of hours, body totally get into contraction because of lack of ATP. So ATP is needed to break that contraction. And that means muscle has to make ATPs whenever it is contracting. It has to keep ATPs to break it. And for that, it has to keep metabolic pathways running. And that's why calcium will have a positive effect on pyruvate dehydrogenase complex, where pyruvate is converted to acetyl-CoA, and TCA cycle also, key enzyme in TCA cycle, positively modulated by calcium. We are going to see that today. So both ADP and calcium will make sure metabolic pathways runs faster. Is it clear? Okay, this is how you need to remember regulation of metabolic pathways. The application, clinical scenario or daily life scenario, put it there. Okay, so there is covalent modification and hormonal regulation, both go hand in hand. Let's look at our insulin. What is the role of insulin on your blood glucose level? Decrease blood glucose. How it will decrease blood glucose? It will act on PFK1 indirectly through PFK2, increase fructose 2,6-bisphosphate, keep your PFK1 active, and then glycolysis goes faster and you get pyruvate. So insulin has taken care to bring glucose all the way to pyruvate. So it's not going to stop there. What insulin do? It will keep your PDC in dephosphorylated state. That means what insulin do? It's going to activate protein phosphatase, just like what I said earlier. And that protein phosphatase removes if there is any phosphate over the surface of PDC and keep the enzyme active. And once your PDC is active, so it's going to oxidize pyruvate into acetyl-CoA. Okay, and that is what I have written here. Insulin causes dephosphorylation of PDC. That's the covalent modification. And that's an active form of enzyme, which I have written here. And then what glucagon has to do? Glucagon does the opposite. It doesn't want glycolysis to go on faster because it wants to save glucose. So it will target all these key enzymes and inactivate them. Inactivate in the sense they keep them kind of inactive, right? So it causes phosphorylation of PDC and keep it inactive. So this is how hormonal and covalent modification goes. So the covalent modification is dependent on the hormonal action. Insulin causes dephosphorylation, glucagon causes phosphorylation. Dephosphorylated form of PDC is active. You got to remember that way. Opposite of that is inactive, done by glucagon. Okay, that's how the regulation of PDC goes. So there can be questions on this. What if there is a defect in E1 subunit of PDC? Let's look at only the E1 subunit of PDC, because each one of our complex enzymes, 3 complex enzymes, they have their own E1 and E2 subunit genes. What we are looking at here in congenital lactic acidosis is mutation in only E1 subunit gene of PDC, not other things. Only PDC E1 gene. That means what happens to your PDC? PDC down. That means pyruvate is not going into acetyl-CoA, it just goes into lactate. So patient will show severe lactic acidosis. And you can see that at the time of birth. That's why it is called congenital lactic acidosis. It can be because of defect in E1 subunit gene, or it can be defect in ETC or TCA cycle enzymes, other causes. But the most likely cause is the E1 subunit gene. That's why I written that first. Okay. And then decreased oxidation of pyruvate into acetyl-CoA into carbon dioxide. So patient will show elevated lactate and decreased blood pH. Basically, it's acidosis, metabolic acidosis. And the consequences, some of the consequences of metabolic acidosis I have written here, like hypotonia, tachypnea, developmental delay, seizures, respiratory issues, motor and intellectual abnormalities. So those are a couple of signs. Just to keep you aware that if there is a defect in PDC, patient will show lactic acidosis. If it is seen at the time of birth, it is a congenital lactic acidosis, and the common cause for that is E1 subunit mutation for PDC. Okay, so that is the thing. Let's look at the question. All of you take out your mobile phones. So this is a question on regulation. So when it comes to regulation question, you got to slow down. Okay, don't jump into conclusion. Slow down, go back to that slide, think about it, and then go to the answer. So this person is running marathon. So what is going on in his muscle? So muscle contraction. Since we are in PDC, so what will happen to your PDC when a person is running? It has to be up, right? It has to be active. And who is going to keep it active? It is the calcium. Remember, calcium. Calcium is a positive modulator of pyruvate dehydrogenase complex. So that's why the option D here, calcium should be the answer. Look at option A. Option A, NADH is it is a feedback negative effect, not activation. It is an inactivation thing, basically. Alpha ketoglutarate dehydrogenase complex, we have not yet covered, and NAD doesn't have any effect on that. We will see it. Increased feedback activation of pyruvate dehydrogenase by oxaloacetate. Some, like one of you probably went for this. It's not oxaloacetate which has a feedback effect. It's acetyl-CoA which has a feedback negative effect.